live cell imaging. Here is a cartoon of our cell shape. Our cell has plasma membrane, also nucleus inside, and mitochondria, lysosome, ER, Golgi kind of structures. So this is how we describe our cells. If you want to visualize specific protein in the cell, you can choose your target protein, and then you can generate the antibody for the protein. And among many antibodies, you can select out the best binding antibody through cloning process. Then you can get very strong binder monoclonal antibody, and then you can label the antibody with a fluorescence motif. Then in this case, your probe is this antibody, and then this is your target or your biomarker. If your biomarker is expressed on cell surface, this is a special type of a biomarker we call CD, cluster of designation. In this case, the antibody can bind to the CD, then you can visualize your target protein. If your biomarker is inside the cell, then even though you have antibody, the antibody cannot be used for the live cell imaging. Because the antibody is a huge macromolecule, it cannot penetrate the live cells, the membrane. In this case, you need to fix the cell first. Here the fixing means chemical treatment utilizing formaldehyde, and then it can connect the amine groups between different protein and make a cross-linking. That is a fixing. Still the membrane is intact. Membrane cannot get in. Now you need to add a detergent and dissolve some part of a membrane, the structure, and then the antibody can go in, can bind to the target. Through this process, the antibody-generated image we call immunofluorescence. For example, if you add anti-tubulin to the muscle cell, here you can see the blue color is the nucleus of the cell, and then the green color is a microtubule structure. So muscle is a multicellular structure connected by microtubules. If a microtubule acting compound myosin is added, then we can see those microtubule structure is fragmented. If you add myosin antibody, you can see different structures. Intact cell shows a similar tubular structure, but myosin-treated cells shows quite unique the structure, which looks quite different from tubulin structures. So this is a typical example of antibody-based imaging. They look quite lively, but these cells are the fixed and dead cells. Our eyes can see multiple colors. So in this case, the green color shows microtubule structure, red color shows actin structure, and blue color representing the nuclear structures. Now, if you want to get live cell imaging, we need to think about the live cell's property. In live cell, most important thing is through the metabolism and also the material exchange, live cells membrane, they have a membrane potential. Usually we consider outside of the cell is positively charged and inside the cell is negatively charged. But if you look at the structure, the chemical structure of a phospholipid, many phospholipids, they are negatively charged. Some of them, they are negative positive. It's a neutralized neutral molecule, but actually there is no positive charged the phospholipid. Then how can you get this positive charge on the outside? Well, inside and outside lipids, they have a different composition of a phospholipid, but in reality, inside is more negative than outside. That's why relatively outside is positive the voltage, but the surface, both sides, is actually negatively charged. That means if we add the negatively charged molecule, this molecule has a problem even to access to the cell surface. Because of this, the electrorepulsion, this molecule has the bad property for the cell penetration. If it is positively charged, it may be beneficial to access to the cell surface. If it is a neutral molecule, then it doesn't have that kind of a charge other problem. Now, next step is crossing the membrane. The cell membrane is about 4 nanometer thickness, which is quite big distance considering the size of the molecule. And here we can consider the Lipinski rule, and C log P value represent the hydrophobicity of the molecule. The higher number means more hydrophobic. So if the molecule is too hydrophilic, so let's say less than 2, then the molecule can be bounced from the surface. If the molecule is the C log P value is bigger than 5, that's too hydrophobic. If it meets any kind of a membrane, it can be stuck to the membrane and doesn't move. So only when the molecule has in between 2 and 5 of a C log P value, then they have a chance in and out and then penetrate the cell membrane, the space. By controlling the chemical properties of a small molecule, we can label the different organelles selectively. If the molecule has a positively charged and reasonable hydrophobicity, they tend to accumulate in mitochondria, because mitochondria has much higher membrane potential comparing to the plasma membrane. And the membranes outside the plasma membrane is the thickest, and then Golgi and EL membrane getting thinner and more flexible. And the molecule is the neutral, and also if it has reasonable hydrophobicity, then depending on their hydrophobicity, they can selectively bind to Golgi or EL, but usually they tend to be fuzzy the staining, because this is only rely on their physical differences. If the molecule has a very high strong binding partner, such as the DEPI or the waste molecule, which bind to the DNA, then by chemical equilibrium, they can be accumulated inside the nucleus. This is a nucleus staining by waste. Waste is a blue dye and also has a strong binding affinity to the nucleus, and it can stain live cells and dead cells and very powerful nucleus dye. So if you have any kind of the fluorescence cell images, maybe more than 90%, if you see the blue color, it would be waste stained the nucleus imaging. If the dye has a reasonable hydrophobicity and also positively charged, then usually they tend to bind to mitochondria. ER staining can be achieved by reasonable hydrophobicity or ER selective binding motif over sulfonamide structures. Cell mask has been used for cell membrane imaging. This is a commercial dye, and unfortunately, the chemical structure is not known. Lysosome usually showing this kind of the punctated structure. And here is a typical example of a lysotracker. This lysosome has acidic environment, and this kind of a basing motif deliver this molecule into this lysosome area. Inside the lysosome, this basing motif can be protonated and is charged, so it doesn't come out from lysosome, and this is the mechanism of lysotracker. Sometimes we need to see live or the dead state of the cell. Here is an example of a live dead the staining kit. Propiondium iodide is a DNA binder with a red color, but this dye itself cannot penetrate the cell membrane, so it can stain only damaged cell membrane damaged cell, so it's kind of a dead cell staining reagent. Live cell staining can be done casein AM ester. AM ester blocks this dye, and the neutral molecule can go inside the cell, and the esterase remove these protecting groups, and then negatively charged the product, green color, filled up the cytosolic area, and if the cell membrane is intact, they cannot come out. So that's why green color here is the live cells, red color is dead cells. Inside the nucleus, there are smaller structures so-called the nucleolus. Nucleolus is the center of ribosomal RNA synthesis. That's why that area has a high contents of RNA. So it was found that RNA selective dyes can have a potential to stain this kind of the nucleolar structures. Not only the physical properties of organelle structure, sometimes the metabolite or signaling molecule such as calcium can also be imaged by indicator or probes. Here is the historically very famous fluoride, which can sense calcium changes inside the cell. Calcium is a third messenger through the signal transduction pathways, and this fluoride's the role, it opened up the whole field of signal transduction. That's why this single paper has been cited more than 20,000 times. This is an amazing number. Well, this eye was developed by Professor Roger Chen. I personally believed he may get the Nobel Prize by this discovery, but he got Nobel Prize, but with a different achievement.